go ahead and start the presentation off with prophase. At prophase, there are two very distinct things that happen. Replicated chromosomes containing two very closely associated sister chromatids condense, which just basically means that the chromosomes are starting to, the chromatids are starting to form, and the mitotic spindle assembles between the two centers. MCDK is a very important protein kinase that affects a lot of the processes during mitosis. Some of MCDK's roles include inducing the assembly of the mitotic spindle, triggering chromosome condensation, the two most important things that happen during prophase, and in the later end of prophase, it also promotes the breakdown of the nuclear envelope. This all occurs when when MCDK phosphorylates specific proteins in each process. This MCDK is also aided by aura kinases and polo-like kinases. Onto the third slide. The dephosphorylation activates MCDK at the onset of mitosis. So this activation of MCDK begins with the accumulation of M-cyclin. An increase in M-cyclin protein leads to a corresponding accumulation of MCDK. At the end of G2, when there's a stockpile of this, MCDK is ready, but it is not ready to act because it is blocked by the phosphates. Then CDC25 removes the inhibitory phosphate while the kinase WEE1 activity is suppressed, ensuring that the MCDK activity is increased. CDC25 can also be activated by its target, which is MCDK. In this process, WEE1 can be inhibited by MCDK. These also suggest that MCDK activation in mitosis is involved in feedback loops, as you can see in the next slide. Partial activation of the CDC25 leads to partial activation of the MCDK. The MCDK molecules then phosphorylate more CDC25 and WEE1, leading to more MCDK activation. In this picture, it shows that the M-cyclin is inactivating the MCDK, and there's both positive feedback loops which activate more MCDK production. As far as the chromosomes go, condensin helps configure the duplicated chromosomes for separation. At the end of S phase, long DNA molecules are tangled in a huge pile of mess, kind of like string in a big knotted ball. Just pulling these apart would lead to breaks in the chromosomes and in the DNA structure, which we wouldn't want because then it would be a mess. So they go through two processes to untangle themselves, chromosome condensation and then sister chromatid resolution. During chromosome condensation, it is dependent on the 5 subunit protein called condensin, and the condensin structure, as seen below, is related to cohesin structure. The condensin may form a ring-like structure in the blue that uses energy from ATP hydrolysis to promote compacting and resolution of the sister chromatids. So basically it allows the DNA to loop and to continue looping to form the condensed structure of that chromatid. Going on, the mitotic spindle within prophase is a microtubule-based machine. The mitotic spindle is a bipolar array, which means two separate areas of microtubules that pull sister chromatids apart in anaphase, but they are put together here in prophase. They segregate the two sets of chromosomes to opposite ends of the cell. MCDK, of course, triggers the assembly of the spindle during early mitosis, and it occurs while the chromosomes are configured, as just discussed in the two processes. The mitotic spindle, again, is a bipolar array of microtubules, as seen below. The minus ends are focused at the spindle poles, and the plus ends radiate outward from the poles. So the plus ends are more towards, I guess, the nucleus of the cell, and the minus ends are located more toward the spindle poles. Moving on, the plus ends are called interpolar microtubules. They interact with the plus ends of microtubules from the other pole, seeing as both positive ends meet in the middle, and they result in an antiparallel array in the spindle midzone, kind of where the chromosomes are. Kinetochore microtubules are another type. They attach to sister chromatid pairs at the large protein portions called kinetophores, and they're located at the centromere of each sister chromatid. So it's kind of that central location where they're held together is where the kinetochore microtubules attach. And there are also